you have a network that is formed between parallel channels or among parallel channels, so that the parallel channels are no longer independent, such that they are talking to each other. And that is additional level of computation which has been speculated but never been directly proven like Yao did.
So my name is Yao Xue. I was a PhD student, now a postdoctoral scholar in Dr. Jimmy Zhou's lab. And this paper is basically my thesis study. So our lab, Zhou lab is interested in studying the retinal circuitry, how the retina process the incoming light patterns and send the signals to the brain to form our conscious vision. So my study's subject is the retinal bipolar cell, which is a class of interneurons within the retina that relay the signal from the photoreceptor, like rod and cones, to the downstream ganglion cells or amacrine cells. So we are very interested in how these different types of bipolar cells process the information, because there's not only one type, just one type of bipolar cell in the mammalian retina, or specifically in the mouse retina. There are actually over 15 types there. So it's a huge number. And each type, people think that they perform a little bit different task. So my study is basically elucidating how they perform the uh, visual processing, and then how they extract the different visual features there. And then what we surprisingly found is that they don't just very separately and independently process the information, but rather they use a very special circuitry to do a coherent processing there.
Hi, I'm Seung-hoon, and I'm a colleague of Yao, and I work in Dr. Zhou's lab. And like Yao, I have interest in studying some interactions between neurons. So for this Yao's project for bipolar cell, I kind of try to aid Yao's experiments, and then participate a lot of discussions for analyze or some analysis of data. Basically, in our lab, we try to understand using physiology, and recently adding some other connectomic studies using some AI guided connectomic studies, try to understand from very anatomical point of view to the functional point of view of the cell in the retina, and then we try to understand how these neurons to act, perform during the certain visual processings. So that's our kind of usually job in our labs we are doing.
So this paper is to study interneurons in the retina, particularly as Yao and Seung-hoon already alluded to, interneuron called bipolar cell, which is the second order neuron in the retina. There are photoreceptor cells, which are the first order neuron accepting light. They're receiving light, so they're called photoreceptor cells. And then the second layer is called bipolar cells, which is bipolar, meaning it receives signal from the photoreceptors at one pole, and the other pole they release information onto the output neuron of the retina. So the bipolar cells have there are 15 types or around that number in the mouse and in most of the mammalian species. It has been generally believed that those 15 or so types of neurons, they form parallel independent processing unit, such that when signals are received by the retina, they are divided into parallel channels for parallel visual processing. And that's fundamental because it's believed to be a process that go from the retina all the way to the visual cortex. So the supporting evidence for that is the synaptic connectivity. That is, neuron to neuron connections are formed by synapses. So typically, when we say synapses, people usually refer to chemical synapses. So a presynaptic neuron will release neurotransmitter, and which will be received by the postsynaptic neuron. So based on chemical synaptic connectivity, it's believed that those channels are more or less independent and separate. So what Yao found is that in addition to the chemical synapses, there are additional electrical synapses. Electrical synapses are known, it's not new, but it's hard to detect because they're much smaller, cannot be easily seen under microscope. So what Yao found is that because of the chemical synapses, which are formed between bipolar cells, then you have a network that is formed between parallel channels or among parallel channels, so that the parallel channels are no longer uh, independent, such that they are talking to each other. And that is additional level of computation which has been speculated but never been directly proven like Yao did. So with this finding, Yao and Shen Hong discovered that signals can be processed by the whole network instead of a single channel. And then even among the channels, there are driver channels and the passive channels. In other words, there are channels that are playing more important role, and through the gap junction, they can drive the other channels. So those are the fundamental discoveries in the visual system. This is the first stage of visual system, which is in the retina, which is actually the first synapse in the visual system, from the photoreceptor cells to the bipolar cells. And along the way, we also used, well, this was first done in mice. And then we tried to prove this has a general implication to many species. We looked at human retina. And with the help of Machara, uh, who is the pathologist here and who provided us with fresh human retina. Fresh meaning 2 to 3 hours postmortem, which is very difficult to obtain. And then because of Yale's program called the Legacy Donor Program, which Machado can describe to you in more detail. And through this program, and Yao was able to record from postmortem human retina. This was the first time, I think, to be recording using electrophysiology in the whole mount human retina, human postmortem retina. And it was surprising to find that even after 2 hours of postmortem period, the neurons in the retina are still connected and they're still functional. And then using this similar approach, Yao showed that similar processes are going on in the human retina as well.
The Yale Legacy Tissue Donation Program is a rapid research autopsy program that we run here at Yale. Um, and there are a few other rapid autopsy programs around the country, but the one here at Yale is unique in that we are not dedicated to one particular disease or one particular organ system. And so what that allows us to do is enroll a very heterogeneous sort of cohort of donors, and that provides really great material for research for basic physiology, right? So a patient who is being seen for a primary cardiovascular disease may have a relatively healthy central nervous system and eyes, for example, and therefore provide really good material for research into sort of basic normal physiology. And so that is something that really is it's driven by tremendous generosity of our donors and their families. We like to say that, you could have all the funding in the world, but there's no substitute for, the material to study. I mean, actually, the retina is a good example. Retinal circuitry is known to be, to vary among species. There's tremendous evolutionary difference among, different species in the particulars of retinal neuronal circuitry. And so, with this, really exciting finding of integrated processing in bipolar cells in mice, it's certainly not given in that functionality is conserved up through human, and whether that actually plays a role in us or whether the circuitry is arranged differently. But by basically enabling the sort of same type of confirmatory experiments, i.e., in um, live physiology, we, along with Dr. Zhao's lab, are able to, show that those mechanisms are indeed conserved in the human eye. So I think it was a really exciting, collaboration and use of our material.